Hello, dear students, welcome to our today's program. Today we are going to discuss biochemical analysis of body fluids. Today we'll discuss blood and urine, specimen collection and processing. The main objectives of today's lecture are to know the composition of blood, to know the composition of urine, knowledge about various biochemical tests of blood and urine, specimen preparation for various biochemical tests. Now let's have a discussion about blood. Blood is a special connective tissue consisting of a fluid matrix, plasma and formed elements. Now let's discuss first plasma. Plasma is a straw-colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water and proteins contribute 6 to 8% of it. Fibrinogen, globulins and albumins are the major proteins. Fibrinogens are needed for clotting or coagulation of blood. Globulins primarily are involved in defense mechanisms of the body, and the albumins help in osmotic balance. Plasma also contains small amounts of minerals like sodium, calcium, magnesium, bicarbonate, chloride, etc. Glucose, amino acids, lipids, etc. are also present in the plasma as they are always in transit in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without the clotting factors is called serum. Now let's discuss formed elements. Erythrocytes, leukocytes and platelets are collectively called as formed elements, and they constitute nearly 45% of the blood. Erythrocytes or red blood cells, also called as RBCs, are the most abundant of all the cells in the blood. A healthy adult man has on an average 5 million to 5.5 million of RBCs per cubic millimeters of blood. RBCs are formed in the red bone marrow in the adults. RBCs are devoid of nucleus in most of the mammals and are biconcave in shape. They have a red-colored iron-containing complex protein called hemoglobin, hence the color and name of these cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. These molecules play a significant role in the transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen, which is also known as the graveyard of RBCs. Dear students, now let's discuss leukocytes. Leukocytes are also known as white blood cells or WBCs, as they are colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which averages 6,000 to 8,000 per cubic millimeter of blood. Leukocytes are generally short-lived. There are two main categories of WBCs, granulocytes and agranulocytes. Neutrophils, eosinophils and basophils are different types of granulocytes, while lymphocytes and monocytes are agranulocytes. Neutrophils are the most abundant cells of the total WBCs and basophils are the least among them. Neutrophils and monocytes are phagocytic cells which destroy foreign organisms entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reactions. Platelets, also called thrombocytes, are cell fragments produced from the megakaryocytes, which are the special cells in bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets per cubic millimeter. Platelets can release a variety of substances, most of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to the clotting disorders, which will lead to excessive loss of blood from the body. Now let's have a discussion about how do we collect the blood. Venous blood is collected usually from antecubital vein or some other prominent veins of the forearm under aseptic conditions. arterial blood is required rarely. This may be collected from radial, brachial, femoral artery. Capillary blood may be collected from the tip of the thumb or finger or from the earlobe. Now let's discuss some experiments regarding the blood. Centrifuge a sample of oxalated blood. Observe that cells are sedimented and plasma is separated. Normally the color of the plasma is pale yellow. Add 10 drops of 2.5% of calcium chloride solution to the 10 ml of oxalated blood. Mix and let it stand. The blood clots. Oxalate removes it as insoluble calcium oxalate, preventing clotting. When additional calcium ions are added, clotting occurs. Keep the clotted blood for an hour. A fluid separates while the clot retracts. Transfer the fluid to a centrifuge tube and briefly centrifuge. The clear supernatant is serum. Note its color. Normally, it's the light yellow. Plasma and serum are chemically same except that the serum lacks fibrinogen. Now let's discuss the test for proteins in blood. First, globulins. To 2 ml serum, add 2 ml saturated ammonium sulfate solution, which is also known as half saturation. The globulins are precipitated. Filter the solution. Test the filtrate by biuret reaction using 40% sodium hydroxide solution. If test is positive, then filtrate contains serum albumin that was not precipitated by half saturation. The next is albumin. Fully saturate with ammonium sulfate crystals the filtrate of the above experiment. Precipitation will be observed, which is due to albumin. Next, we'll discuss fibrinogen. Mix 0.5 milliliters of plasma, 15 ml water and 0.5 ml of 2.5 calcium chloride solutions in a small beaker. Allow it to stand for 20 minutes at 37 degrees Celsius in the incubator. After incubation, insert a tapered glass rod into the solution and feel the transparent clot formed and collect it by pressing against the walls of the beaker. The clot is fibrin, the insoluble form of fibrinogen. Suspend the fibrin clot in normal saline to remove the adhering proteins and dissolve in 5 ml of 5% sodium hydroxide. Perform biuret test with the solution. It's positive. Fibrin is a protein. Dear students, now let us have a look for the test for inorganic constituents in the blood. First, deproteinization of serum. Take 10 ml serum in a test tube, add a few drops of 2% acetic acid, keep in boiling water for 5 minutes. The proteins coagulate, then filter it. The filtrate is the protein free of serum containing all the inorganic constituents except proteins. This filtrate can be used for many experiments. Let's discuss the first one, test for chlorides. To 1 ml of this filtrate, add 2 drops of concentrated nitric acid and 2 drops of 3% silver nitrate. A white precipitate of silver chloride shows presence of chlorides in serum. Now, this test is based on the principle that chlorides react with silver nitrate, forming a white precipitate of silver chloride. Nitric acid prevents the precipitation of salts other than chlorides. Next test is for the test of phosphates in blood. To 2 ml of the already collected filtrate, add 2 ml of ammonium molybdate reagent and a few drops of concentrated nitric acid. Warm, if necessary, to get a canary yellow color, which is due to the presence of phosphates in the serum. Now, this reaction is based on the principle, in the presence of nitric acid, phosphorus reacts with the ammonium molybdate to form a yellow precipitate of ammonium phosphor molybdate. Moving on to the next test, it's test for calcium. To 2 ml filtrate, add 1 ml of saturated ammonium oxalate. A white cloudiness is due to the precipitation of calcium oxalate, which indicates presence of calcium. Now, this test is based on the principle that ammonium oxalate reacts with the calcium to form a white precipitate of calcium oxalate. Test for organic constituents in the filtrate. First being test for glucose in blood. 0.5 ml filtrate, add 1 ml of Follen's alkaline copper sulfate solution, mix and keep in boiling water for 5 minutes. Cool it. Add 5 ml of phosphoromolybdic acid reagent. A deep blue color shows the presence of glucose. Next is test for urea. To 0.6 ml of filtrate, add 0.2 ml of aqueous horse gram suspension, which is 10%. Mix and keep in warm water for 10 minutes. Now add 5 ml water, mix and filter. To the filtrate, add 2 ml of Nessler's reagent. A brownish yellow color indicates presence of urea. This test is based on the principle that the enzyme urease present in the horse gram converts urea into ammonium carbonate, which gives color with Nessler's reagent. Now let's discuss the test for uric acid in blood. To 0.5 ml filtrate, add 1 ml of 10% sodium carbonate and 1 ml of phosphotungstic acid, dilute Follen and Dennis. A blue color develops due to uric acid. Next, let's have a look at the test for creatinine. To 1 ml filtrate and 0.5 ml of 1% picric acid followed by 0.5 ml of 10% sodium hydroxide. The yellow color changes to orange due to the presence of creatinine. This is also known as Jeffreys reaction, which is quantitative for the estimation of creatinine in serum and urine. Now let's discuss chemical analysis of urine. Urine is an excretory product of the body, and presence of certain substances in the urine reflects the metabolic state of the body. Since it can be easily collected and examined, routine and microscopic examination of urine are preliminary and important in diagnosis of various pathological conditions. Let's have a look at collection of specimen and its preservation. Like all biological specimens, urine has to be collected and adequately preserved to prevent contamination and bacterial overgrowth, since it's a very good culture medium. The type of urine specimen to be collected is determined by the test to be performed. First, a clean, early morning fasting specimen is generally the most concentrated specimen and preferred for microscopic examination and for detection of abnormal amounts of constituents, for example, proteins. Second, a clean, timed specimen is one obtained at specific times of the day or during certain phases of the act of micturition. First, 10 ml of urine, voided is most appropriate to detect erythrocytes. Midstream specimen is best for bacteriological study. Third, catheter specimens are used for microbiological examination in critically ill patients or in urinary tract obstruction only. Now let's discuss the preservatives used. The most satisfactory form of preservation is refrigeration at 40 degrees Celsius combined with chemical preservation. Commonly used forms of preservation used earlier were formalin, 2 drops of 40% in 30 ml of urine, or thymol, 0.1 milligrams per 100 ml of urine sample. Nowadays, tablets containing a mixture of chemicals are widely used. They act by lowering the pH and by releasing formaldehyde. For ketone bodies, investigation is to be done immediately or within 2 hours of collection, or it should be refrigerated with adequate preservative. Now let's have a look at physical examination of urine. First being color. Normally, the urine is colorless to straw colored due to urochrome. Deep yellow, mild to severe dehydration, jaundice, B complex therapy due to riboflavin, red to brown, hematuria, hemoglobinuria, myoglobinuria, porphyria, erythropoietic type. Brown to black, alkaptonuria, methemoglobinuria. Now let's discuss the appearance of urine. Normal urine is perfectly clear and transparent when freshly voided. It may become turbid if exposed for a long time due to the urea being acted upon by bacteria and converted into ammonium carbonate, or due to separation of mucoproteins. Turbidity in urine. Phosphate excretion in alkaline urine pus cells. Specific gravity is measured by urinometer, implies the capacity of kidneys to concentrate urine. Normal value 1.002 to 1.028. It depends upon state of hydration and solute load. Values more than 1.028 imply severe dehydration. For example, in diabetes mellitus, adrenal insufficiency. Values less than 1.002 indicates increased water intake. For example, in diabetes insipidus and chronic nephritis. Volume of urine. Normal value 700 to 2000 milliliters per day. Depends upon fluid intake, solute load, loss of fluid by skin or otherwise. Climatic conditions. Now let's discuss some important terms. Polyuria, more than 3 liters per day of urine. Conditions: diabetes mellitus, diabetes insipidus, recovery from acute renal failure ARF, diuretic therapy. Oliguria, less than 500 milliliters per day of urine. Conditions: ARF, vomiting, fever, burns. Next is anuria, less than 50 ml per 12 hours. Now let's discuss the pH of urine. Normal range is 4.5 to 8.5. Average 6.0 in 24-hour sample. pH 8.5 or more found. After heavy meals, protease infection, pH 4.5 or less is found. After heavy exercise, metabolic acidosis, chronic respiratory acidosis. Now let's discuss chemical examination. First, proteins. Normal adults excrete up to 150 milligrams proteins per day. Nephrotic syndrome is a clinical condition when kidney loses more than 3.5 gram proteins per day per 1.73 meter square with hypoalbuminemia, edema, hyperlipidemia, lipiduria and hypercoagulability. Microalbuminuria, a condition characterized by urinary albumin excretion rates between 20 to 200 micrograms per minute or 30 to 300 milligrams per 24 hours. This is shown to precede the overt renal disease and is an indicative of increased risk for development of diabetic nephropathy. Second, glucose. Normal urine has reducing sugars in the concentration of 1 to 1.5 grams per liter. Of this, glucose is 200 to 300 mg. Glucosuria, glucose in the urine beyond the normal range. Renal threshold. In normal persons, so long as the blood glucose is less than 160 to 180 milligrams per deciliter, glucose is not excreted by the kidney in amounts which are detected by the routine tests used. This level is termed the renal threshold for glucose. Renal glycosuria. It's a condition in which the renal threshold for glucose decreases so that the glucose is present in urine in the presence of normal blood sugar levels. Physiological renal glycosuria is seen in pregnancy. Third, ketone bodies. Ketone bodies are beta hydroxylated butyrate 78%, acetoacetate 20% and acetone 2%. Normal blood levels 0.5 to 1.5 mg%. Normal amounts in urine are 50 mg per day. ketonuria seen in diabetic ketoacidosis, starvation, severe vomiting, glycogen storage disorders, high-fat diet. Fourth, let's discuss blood in urine. Presence of RBCs in urine is called hematuria. Hematuria seen in nephrolithiasis, malignant hypertension, sickle cell anemia, coagulation abnormalities. Fifth, bilirubin. A freshly voided specimen is required since bilirubin is very unstable. Unconjugated bilirubin does not cross the glomerular basement membrane as it is non-polar and water insoluble. Conjugated passes freely as it is water soluble. Sixth, urobilinogen. It's a colorless compound formed in the intestine by the action of gut flora on bilirubin and is reabsorbed by enterohepatic circulation and excreted in urine. Dear students, let's discuss microscopic examination. Cells. In these RBCs, WBCs or pus cells, epithelial cells or normal desquamation from urinary tract, bacteria if any. Number 2, crystals. Associated with renal calculi commonly is oxalate in acidic urine and phosphate in alkaline urine. Number 3, casts. These are masses of agglutinated proteins in the form of cylindrical moles of tubular lumen. Well, dear students, I really hope you would have understood today's lecture. Thank you.